The THEA-Code is an autoencoder-based IDS-correcting code for DNA storage. It addresses the challenges of DNA storage by employing an end-to-end autoencoder for encoding and decoding processes. The code introduces innovative techniques such as the differentiable IDS channel, entropy constraint on the codeword, and auxiliary reconstruction of the source sequence. These strategies contribute to the successful convergence of the autoencoder, resulting in a deep learning-based IDS-correcting code with commendable performance. The THEA-Code is the first instance of a deep learning-based code independent of conventional coding frameworks in the IDS-correcting domain. Comprehensive experiments, including an ablation study, provide a detailed analysis and affirm the effectiveness of the THEA-Code. The research paper discusses the development of a novel deep learning-based coding scheme, dubbed the THEA-Code, which addresses challenges in integrating deep learning methods with coding theory. The scheme employs a differentiable IDS channel, an entropy constraint on the codeword, and auxiliary reconstruction of the source sequence. The differentiable IDS channel, implemented using a sequence-to-sequence model, simulates conventional IDS operations, allowing for gradient propagation. The entropy constraint on the codeword ensures that the probability vectors used by the decoder are close to one-hot vectors, addressing information loss from quantization. The scheme's optimization target includes a cross-entropy loss for reconstruction disparity and the Kullback-Leibler divergence for training the differentiable IDS channel. The paper highlights the significance of these novel approaches in enhancing the performance of deep learning-based coding schemes. The research paper discusses the development of a novel DNA data storage system, focusing on the encoder and decoder's roles in sequence-to-sequence models. It introduces an auxiliary reconstruction task for the encoder to ensure it can replicate the input sequence without reduction. This task, combined with the entropy constraint and the auxiliary loss, enhances the model's ability to handle the chicken-and-egg dilemma during training. The encoder and decoder are implemented using transformer-based sequence-to-sequence models. The training phase consists of two parts. First, training the differentiable IDS channel, and then training the autoencoder with the weighted sum of the cross-entropy loss, entropy constraint, and auxiliary reconstruction loss. The testing phase involves replacing the differentiable IDS channel with a conventional one and evaluating the performance using metrics such as the nucleobase error rate, NER. The paper presents an ablation study, showing the performance of the proposed method against different source lengths and coder rates, highlighting its effectiveness in handling various scenarios. The research paper explores the use of deep learning models for error correction in DNA storage systems, focusing on the design of a novel autoencoder that can correct insertion, deletion, and substitution errors. The authors demonstrate that by optimizing the hyperparameter lambda, which dictates the weight of the entropy constraint, the autoencoder's performance can be significantly improved. They also investigate the effects of the auxiliary reconstruction loss and its impact on the overall performance of the model. In their experiments, the authors observe a trend where the NER, nucleotide error rate, increases from 0.36% to 6.07% along with the enlarged coder rate. This suggests that an autoencoder can maintain its performance even at higher coder rates, where the burden on the deep model is relieved. Furthermore, the results indicate that an extra 6.70% NER is introduced by the IDS errors in the codeword when using a coder rate of less than 80%. The paper also presents an analysis of the hyperparameter lambda, which controls the weight of the entropy constraint and the overall loss. The authors find that careful tuning of this parameter is crucial to achieving optimal performance. Over-applied entropy constraints can lead to a local minimum convergence point, while under-applied constraints may result in codewords that are diverse from the one-hot style, altering the input domain for the decoder and failing the testing phase. Additionally, the authors investigate the effects of the auxiliary reconstruction loss on the model's performance. They observe that the reconstruction loss LCE between the source and sink sequences converges well when using lambda equals 0.01, and the entropy maintains at a low level. This verifies their conjecture that an over-applied entropy constraint will lead the model to a local minimum convergence point. Overall, the paper demonstrates the potential of deep learning models for error correction in DNA storage systems, highlighting the importance of optimizing hyperparameters and auxiliary reconstruction losses to achieve optimal performance. The paper discusses the application of deep learning techniques to error-correcting codes for DNA data storage. The authors propose an autoencoder-based model that can learn to correct errors in the form of deletions, insertions, and substitutions. This model is trained with an auxiliary reconstruction loss, which imparts a basic logical ability to the encoder. The authors also experiment with different weights for the auxiliary loss and find that it is essential for the model's convergence but does not significantly impact the final performance. Additionally, the authors explore the use of more complex tasks to impart higher initial logical ability to the encoder, such as recovering the sequence from operations like forward difference and position information in coded sequences. They find that the reconstruction of the identity mapping outperforms other combinations, while the combination of identity, forward difference, and position information performs the worst. The paper also reviews existing literature on DNA data storage and error correction, highlighting the importance of robust and efficient storage architectures. It discusses various error-correcting codes and their applications in DNA storage systems, including the use of autoencoders, unsupervised learning, and deep architectures for correcting errors. In conclusion, the paper presents a novel approach to error correction in DNA data storage using deep learning techniques and provides insights into the effectiveness of different auxiliary losses and tasks for initializing the training of the autoencoder. The paper presents a comprehensive overview of the current state-of-the-art in error correction codes for DNA-based data storage, focusing on the use of machine learning techniques to enhance reliability and efficiency. It begins by discussing the challenges posed by insertions, deletions, and substitutions in DNA sequences, highlighting the importance of developing robust error correction methods. The authors then review various existing error correction codes, including puller codes, segmented edit error correcting codes, and synchronization strains. They also discuss the role of machine learning in improving these codes, citing examples such as deep learning-based decoding of puller codes and hypergraph network decoders for block codes. The paper further explores the application of machine learning to create new types of error correction codes, such as codes, which utilize nonlinear encoding and decoding processes. Additionally, it touches upon the potential benefits of using autoencoder-based error correction coding for one-bit quantization. The authors emphasize the significance of information theory in understanding the fundamental limits of error correction and highlight the potential for future research in this area. They also mention the importance of domain generalization in machine learning models for wireless communications, as well as the scaling of deep learning-based decoding via partitioning. Finally, the paper concludes by discussing the potential for future advancements in error correction codes through the integration of machine learning techniques, with a focus on achieving higher reliability and efficiency in DNA-based data storage systems.